Legan is a drink that is produced by local people from the plantation of Siwalan tree and lontar tree. This drink is obtained by the male flower stalk. This produces a liquid with a sweet taste called legan.
In general, nira lontar has a sweet taste that is preferred by the community. It can be consumed directly or processed into various commercial products, such as soy sauce or brown sugar. In Gresik, legan has played a role as an economic driver. This is evidenced by its use in Panjang sub-district and Henders Dairy Village, Menganti sub-district, Gresik Regency, where many people work as palm farmer, using palm trees as food and drink, especially legan, which continues to be preserved. Legan is used to drink and offers many benefits. Legan is mainly sucrose. The phosphorus contained in legan can optimize the excretion process in humans. This causes legan to have good result in treating various diseases such as kidney stones, lungs, tuberculosis, dysentery, defecation, and hemorrhoids.
Kenapa selama ini beli legan?
Karena bisa menyehatkan tubuh.
Setelah ibu meminum legan, apakah penyakit ibu membaik?
Alhamdulillah semakin membaik.
Problem. Despite its usefulness, legan can turn into palm wine. This is a drink with a high alcohol content. Legan can only be kept for less than 24 hours. It will ferment and turn into palm wine. In palm wine, bacteria can be found that use glucose, a source of food, to carry out a metabolic process. In this metabolic process, the acid is converted into a fermentation product in the form of alcohol, namely ethanol and carbon dioxide. The taste is more bitter and sour when ethanol content is higher.
Legan ya takutnya cuma rasanya itu agak berubah, agak asin, terus agak sepet, agak pahit, misalnya.
Due to its bitter taste and high alcohol content, the palm wine drink made from legan cannot be consumed directly, which is not in line with the majority of people's opinion that alcohol is a drink not suitable for consumption. Legan that has been transformed into palm wine is also generally difficult for a consumer to distinguish it directly. This is because the process is relatively quick and difficult to detect effectively. The process of detecting alcohol in legan is generally done manually. A door and taste indicator are ineffective. The sale of legan, which is one of the development in the local economy, may be affected by the lack of effective detection of palm wine and legan.
Waktu saya simpan di ruang suhu biasa, ternyata 1 hari sudah berubah jadi toa.
Objectives A. The aim of this research is to find a way of processing legan with good quality. This will make legan a drink with a longer shelf life and practical. Goals B. The aim of our project is the production of a sticker that will be an indicator for the detection of the alcohol content in legan drinks. The media we use are ingredient proteins or microbes. They are stored directly in a liquid or solid medium. The interaction of alcohol and the media we will use produce gene expression. This can be detected by a pH indicator. The pH indicator will produce a wina. The wina can be the result of detecting alcohol levels according to the color contained in the sticker. Through this project, it is hoped that buyer will be able to monitoring the quality of legan drinks before they become palm wine. This will create product quality and increase product hygiene. Hypothesis A. The hypothesis of this research is to produce powdered ligand drinks. This can be an alternative in the development of the strain. The sweetness of the ligand and the benefit it contains can be maintained by making it into a powder in addition compared to liquid ligand, which can turn into palm wine in less than 24 hours. Powdered ligand is more convenient to consume as it is easy to be stored and processed and has long shelf life. Hypothesis B. The hypothesis of our project is that the bacterium Escherichia coli can be used to induce the production of synthesis ADA derived from Variophorax and synthesized ELDA derived from Actinobacter pyti in larger quantities using recombinant plasmid technology. We use the synthesis of ADH and ALDH to induce the production of pyruvic acid from the alcohol fermentation process. The pyruvic acid produced can be detected by a pH indicator. Methods A. Ligand heating. Ligand as pasteurized before being converted into powder to reduce the level of microorganisms in order to produce better quality and longer lasting ligand. Ligand was first filtered. It was then placed in a pot with water in a 50-50 ratio. The pasteurization process was carried out, with the first pasteurization being carried out using a stove at 62 Celsius for 15 minutes, which was then put into plastic cups using a hot filling system and sealed using a cup sealer at 125 degrees Celsius. The same temperature and time is then used for the second pasteurization. 2. Bleeding process. Maltodextrin with 10%, 15%, and 20% composition was mixed with 5% egg and 100 ml of egg white. This was done to find the best composition of ingredients to form egg white powder. 3. Foaming. A foaming again is used as an additive to form or maintain the homogeneity of a gas-based expression in liquid or solid food, which plays an important role in the foam maturing process. The foaming again used in this study was 5% egg white albumin. 4. Foam drying method. Foam drying is a method of making foam from liquid ingredients with a foam stabilizer added at the temperature of 70 until 75 degrees Celsius, which is then poured onto a baking tray and dried with a blower oven until the solution is dry. This method is an economical way to produce beverage powder and has good drying result. Method B. The method we use is recombinant plasmid technology. This is a technology in the field of molecular biology that uses plasmid to insert a desired gene into a bacterium or microbe that is used for various purposes. 1. Search for GOI, gene of interest. We use the ADA and ALDA genes as a target in other microbes to be engaged to induce the production of acetic acid from alcohol fermentation. Microbes that want to take their genes and engage them have to meet a criteria that can produce ADA or ALDA. In this project, ADA is taken from Variophorax. ALDA is taken from Actinobacter pyti. This research used in CPDI database to get the sequence of gene that will be introduced into plasmid. GOI isolation. Gene isolation allows you to remove unnecessary parts and take the desired genes. Gene isolation is done to make the expected result of molecular genetic injuries more specific. In this research, we simulate the gene isolation by using PCR product features in bleaching.com. 3. combination of GOI and plasmid. Plasmid PAT-28A is used in the study as backbone. Plasmid PAT-28A can be used as bacterial vector for expression. The maximum size of DNA insert for PAT-28A is 4,000 and 1,500. The GOI that used in this research is the ADA with a size of 1.521 pp and the LDA with the size of 1,467 pp. PAT-28A was built with two promoter system that is T7 and lethal promoter. Also, it has canamnicin resistant DNA for selection. The multiple cloning site is located after promoter T7. This study used promoter T7 as GOI promoter. The GOI and plasmid combination process is as follows. A. Restricted cutting. The cutting is carried out at the restriction sites. It is possible to cut the plasmid and insert the desert DNA using restriction enzymes. Plasmid PAT-28 was cut by Sol and Echo 58KL. The cutting site is located on multiple cloning site. B. Ligation connecting. Ligase enzymes are used to splice the GOI and plasmids before ligated the GOI. Restriction site has to be added on the primer of GOI because expression direction of promoter T7 is opposite from the replication direction. Echo 53KL cutting sites add to 5 paginning of ADA gene and Sol cutting site add to ALDS 3N. The study chose PSU 361 enzymes to connect ADA and ALDA gene. PBS was added at the beginning of ALDA gene from increasing the probability of gene expression. C. Transformation. Transformation is the process by which the generic material that has been inserted into the plasmid is combined with the parent's bacterium. In this process, the vector does not join the chromosome of the parent bacteria. Instead, it divides or multiplies independently. Here you can see the project visually by web bleaching. Bellifer sensor system flow. By using the alcoholic fermentation process, the expression of ADA and ADA proteins is produced. This protein trigger the formation of acetic acid so that the amount of glucose concentration fermented by pH indicator is measured. The more acetic acid is produced, the more acidic the pH will be. Result A. The powder can be stored for 3 months. It will be monitored from 30 July 2023 to 27 September 2023. The taste of the powder is sweet and the color is brownish white. The smell of the powder is likely fleecy. It dissolves within 13 seconds and has a pH of 5. Result B. This product has a detector. This is spared from the bottle cap, part of the ligand during product. The bacteria or protein have been stored and sealed in the detector so that the bacteria or protein stored in the detector can be protected from various types of contamination. To use this product, the user can place a sample of drop of the ligand contained in the bottle to be placed on the detector. The detector will experience a color change, which is an indicator of the alcohol content of the ligand drinks. The sticker on the side of the beverage bottle with the pH gradation is used as reference for the level of alcohol content in ligand drinks. The color found on the detector is matched to the color on the sticker on the basis of the reference sticker on the bottle of beverage packing. If the color produced by the detector is incorrectly related to the left area of the sticker, it indicates an acid condition so that it is not safe to drink because it becomes palm wine. Conversely, if the color of the detector shows a color that related to the area that is leaning to the right, then the quality of the lichen is getting better and it's suitable for consumption.